good morning, and thank you very much for that kind introduction. It really is a great pleasure to be here this morning, and I realize the weather has been like California weather for most of the week, so I can't really take credit for it, but thanks so much for arranging that. It's really been a perfect day to be here. I have a long-standing interest in understanding how neural circuits are formed during development. I'm particularly interested in the cellular recognition molecules that neurites, axons, and dendrites express, and how they use that to determine who they should not make synaptic connections with. Now, this is a problem of daunting complexity. You consider the human brain with its, and I don't know how this number was derived, 100 billion neurons, and each neuron making 1,000 connections. And in fruit flies, and I also don't know how this number was derived, there are 150,000 neurons, and they're interconnected by millions of synaptic connections. And where those connections have been analyzed at the level of individual identifiable neurons, one is really struck by the remarkable specificity and reproducibility of these patterns. Now, in a very broad sense, we can think of connection specificity as arising through three basic fundamental processes. First, genetically hardwired mechanisms play an important role in specifying where cells form, what their morphologies look like, and how they make specific connections to other neurons. In a second phase, particularly well characterized within vertebrate systems, is a role for spontaneous activity. So this is activity which is not in response to sensory experience, but is also genetically hardwired. And particularly in higher vertebrates, the experience of an animal with its environment plays a crucial role in further sculpting these specific circuits. Now, over the past 30 years or so, considerable progress has been made in identifying cell surface molecules and the signals that interact with them, that specify and regulate patterns of connections during development. And in a very broad sense, we can think of them falling into three different categories. First, what I would call combinatorial strategies. There's an evolutionary core of cell surface and secreted molecules that play an important role in many different systems and in different organisms, as well as different regions of the nervous system, to specify many different patterns of connections. And these are molecules that many of you are probably quite familiar with: the slits and robos, netrins and their receptors, plexins and semaphorins, and a small group of other proteins that act in different combinations. Second, gradients. And here I'm restricting my comments to gradients of cell surface-bound molecules. And a particularly striking example of that is the role of efferents and Fs in controlling the formation of topographic maps through a complex mechanism, but clearly one that relies upon gradients. And that's particularly well documented in the development of the vertebrate visual system. And third, there's diversity. And by diversity, I mean families of proteins, large families of proteins, which have similar ectodomains, but these ectodomains have different binding specificities, but would have common signaling mechanisms. So one simple example of the kind of molecules one would like to find are molecules that would specify which synapses form based on molecular properties of binding specificities, but they would signal through a common mechanism to indicate that synapses should form between particular types of cells. Now, as a postdoctoral fellow, I was really introduced to this problem. I came from a DNA replication background in E. coli, and I knew nothing about brains. I'd never looked at a fruit fly before I started in Benzer's lab. And I came to Benzer's lab to study learning and memory, and I wrote my postdoctoral fellowship for that funding. And then when I arrived in Benzer's lab, he told me that he was no longer interested in learning and memory, but that he was interested in how neurons would form neural networks and specific neuronal connections. And I can't say I was pleased by that decision, but he sent me to the library and said, why don't you think about what system we should study? He knew fully well which system he wanted to study, and of course I came to the same conclusion. and that was the fly visual system. And two important review articles had a particular impact on me, and they were both written by Ian Meinertzhagen, who continues to work in this field. He was a postdoc at the time, and I particularly like an abstract from one of these reviews in the early 70s, and I'm going to read you a sentence from that abstract, which I think summarizes my thoughts on this system. Still, the optic lobes of the compound eyes, because of their spatially ordered morphology and the iterative nature of most of their connections, offer a system which is one of the least unfavorable. I'd like that, least unfavorable, for the study of the generation of specific neuronal connections. Now, I'd like to argue that this system remains the least unfavorable for studying this problem, and it does so for three reasons. First, we're fortunate to have a fabulous group of markers that allow us to follow the development of different kinds of neurons, as well as literally a complete set of markers for all of the adult neurons within the fly visual system. Second, due to the work of many different laboratories, there's a wonderful set of techniques that allow us to genetically manipulate, for both gain and loss of function, specific cells, individual cells, and analyze them in otherwise wild-type backgrounds. This kind of mosaic analysis turns out to be particularly powerful for analyzing the nervous system. And finally, we're particularly fortunate as we are the beneficiaries of some of the really remarkable work that's been carried out at Genelia Research Campus of HHMI on serial EM reconstruction of the fly brain. And they fortunately use as their test case the part of the brain that we're interested in, and they've been very generous over the years providing us with information as it was being obtained. So it's a marvelous system for studying this question. Now, we weren't the first, and I should say, Meinertzhagen was not the first to focus on the visual system. Cajal was fascinated by the visual system and carried out a series of important studies in the last decade of the 19th century and into the 20th century. And he turned to the fly visual system in his efforts to find a simple system in which to uncover the principles of neural organization and information processing, although he didn't use those terms. And he thought the fly visual system, of course it wasn't the fruit fly, it was a larger dipteran, would be a perfect place to look. And much to his amazement, he found that the fly visual system was of equal complexity to the mammalian visual system. And unfortunately, this was no better place to look for the principles of organization than elsewhere. And this will be the last quote I read to you, but it's from his autobiography about the visual system and has something, obviously it is the source of this title of this talk. The complexity of the insect retina is something stupendous when one considers the inextricable thicket. So that's another term you never see written in a paper these days, the inextricable thicket of compound or faceted eyes. When one penetrates the great retinal segments, one is completely overwhelmed. There I felt more profoundly than in any other subject the shuddering sensation of the unfathomable mystery of life. So we're going to try to uncover some of that unfathomable mystery of life in the next 45 minutes or so. So we became fascinated with this problem of wiring within the visual system, and this is a wonderful diagram from a monograph by Cajal and Sanchez, which does a great job of introducing the system, the photoreceptors in the retina. This is the first optic ganglia that processes some of the initial photoreceptor specific information called the lamina, and the second optic ganglion called the medulla, and then deeper ganglia, which we won't discuss today. And I'll focus on these two parts, the lamina and medulla, in our efforts to understand the molecular mechanisms by which cell recognition molecules play important roles in regulating this process. Now, we've largely used genetics and some not quite genetic approaches to get at this problem. And I'm going to talk about 4 issues today. I'm briefly going to talk about the DSCAM family and its role in regulating dendritic targeting in this first optic ganglion that I described to you, the lamina. In the second part, I'll move to a genetic screen we use. The DSCAM family came from a combination of genetics and biochemistry. NCAD-herin was identified in a genetic screen, which was a behavioral screen, and I'll briefly describe how this general cell adhesion molecule functions. I'll then turn to a family that we've been working on more recently, 2Ig superfamily proteins that interact in both heterophilic and homophilic ways to regulate targeting in the second optic ganglion, the medulla. And then at the end, I'm going to discuss some very recent work that, much to my chagrin, appears to be likely to be playing an important role in fly synaptogenesis, and that's the role of spontaneous activity. We were very proud of the fact that this could all be done without activity, but this may not be the case. So let's go ahead here and move to the first part, which is the initial screens that we did. These were anatomical screens. This is just the adult visual system, the retina of the lamina that's involved in motion detection, and the medulla, and two different neurons that projected different layers in the medulla, the R7 and R8 neurons. And we began with histological screens, and I'll describe one screen very briefly that led to the identification eventually of the DSCAM pathway. And this was a screen that was carried out by Paul Garrity and Young Rao some time ago in the laboratory, and they used pupil lethal mutations. So these were insertions which we knew were disrupting gene activity, p-element insertions, and used, of course, because this would be an easier way of cloning the genes if we found any that were interesting. And these were then screened simply histologically, and this shows a developing third instar visual system. These are the growth cones of the R1 through 6 neurons that terminate in the lamina, and these are the growth cones of R7 and R8 in the medulla. And you can see in this mutant, which they call dreadlocks for obvious reasons, and the acronym being DOC. And here you can see gaps in the lamina and hyperinnervation of the medulla. Now, DOC turned out to encode an adapter protein with 3 SH3 domains and a single SH2 domain. And it seemed very similar in structure to an adapter that by that point in time had been well studied, called RAB2, SEM5, or DRK, depending on what organism you work on. And that was also an SH3, SH2 adapter protein. And it plays a crucial role in coupling receptor tyrosine kinases to the activation of RAS. So by analogy, we simply argued that DOC would act as an adapter between an activated receptor on a growth cone and the analogous G proteins, the small family of P21 GTPases, RAC, Rho, and CDC42 to regulate the actin cytoskeleton. And so using genetics and biochemistry, we identified a group of proteins that play a role in the growth cone, and we turned to a simpler system. It was very difficult to study individual growth cones because at this time there were no good techniques to visualize a single growth cone. So we turned to a simpler system in the embryo, one that had been worked on, indeed had been pioneered by Dietmar Schmucker when he was a graduate student with Herbert Jekla. And Dietmar had come to my laboratory to study the molecular mechanisms of axon guidance, and we began to work on this system in the context of DOC and its interacting proteins. So using genetics, we identified PAC. Through a biochemical approach, we identified this cell surface protein, DSCAM, that acts upstream of DOC. So DOC is an adapter protein to SH3 domains, 2 of them, and an SH2 domain that binds to the receptor. Through the second SH3 domain, it interacts with PAC, and that regulates the actin cytoskeleton. They all give rise to a similar phenotype. They interact with each other in vivo, physically, and also in biochemical experiments. Now, in the process of characterizing the DSCAM gene in more detail, an MD PhD student in the laboratory who was desperately trying to finish his thesis and was working on a very unproductive project that was also related to this, sequenced a bunch of additional cDNAs of DSCAM before we published. We had been only working on one of the cDNAs, one full-length cDNA that we had identified, and he made the remarkable discovery that DSCAM, none of the ectodomains of the 30 cDNAs he sequenced were the same. And very soon afterwards, the complete genome was published, and that made his thesis go very rapidly as he characterized the genomic locus. And that was the work of Wei Shu, Jim Clemens, and Dietmar. So the DSCAM family has 4 different paralogs: DSCAM1, DSCAM2, 3, and 4. DSCAM1 has this crazy alternative splicing giving rise to tens of thousands of isoforms. Each of these ectodomains is characterized by having 3 alternative IG domains, and those 3 IG domains determine recognition specificity. Homophilic recognition specificity requires that all 3 of the variable domains are the same. So that gives rise to an enormous reservoir of interacting proteins in the developing nervous system. DSCAM2 is the kind of gene that molecular biologists should study because there are only 2 alternatives, and the A and B form have different homophilic binding specificity, and DSCAM3 and DSCAM4 are just your standard. By now this isn't standard, but they only have a single ectodomain for each, and all of these show isoform and paralog-specific homophilic binding. In work that I won't describe, my lab and several other labs showed that the DSCAM proteins play an important role in this process called self-avoidance, the ability to recognize self and discriminate between self and non-self, and that's important throughout the peripheral and central nervous systems. And that relies on a process by which each one of these neurons expresses, through a probabilistic mechanism, a set of DSCAM molecules that endows each neuron with a unique identity. DSCAM2 has a related function. It plays a role in tiling. That's the ability of cells of the same class to discriminate between one another, to recognize oneself, and that allows for this smooth coverage of regions of the nervous system, for instance, in the vertebrate and as well as the fly visual system. So there's equal coverage of an entire region for topographic specificity. Now, what I want to focus on today very briefly is that these molecules also act in different combinations. So I'll give one example of DSCAM1 working with DSCAM2 to provide specificity within the lamina, and a second example where DSCAM2 acts with DSCAM4 to provide specificity for another set of dendrites. So the first example is shown here, and here what we're looking at on the left is we're looking at a single unit within the lamina that we call a cartridge, and we're looking at a cross-section through the axon and the neuropil, which is where axons and dendrites are making synapses. The red axons are photoreceptor axons, and all the other guys are interneurons that they make synapses with. Now, I need to remind you, some of you, and then tell the rest of you, because it's widely known, that the synapses of a fruit fly in general look quite different than a mammalian synapse. So the typical mammalian synapse, the typical textbook synapse, is a single presynaptic site and a single postsynaptic element that they release neurotransmitter upon. In flies, to some extent also in worms, but throughout the fly, the standard synapse, if you will, is a single presynaptic site that releases neurotransmitter onto multiple postsynaptic elements, 2, 3, 4, 5, 6, and up. So very complex patterns of connections. The best characterized of these so-called multiple contact synapses is in the retina, and it's called the tetrad synapse, and I just want to focus on 2 parts of the tetrad, so it's a dyad. So here's this tetrad synapse between an R cell, a photoreceptor, which is shown here in red, and 2 of its synaptic partners, 2 lamina neurons called L1 and L2. In all flies, species for which this kind of analysis has been done, there's invariant pairing of dendrites of L1 and L2. And I need to tell you that in each one of these clusters, there's only one L1 and there's only one L2, and each of them have lots and lots of dendrites. Okay? But in all cases, at these synapses, at this there's a dyad, it's always L1 and L2. Both of these neurons play an important role in motion detection. L1 is the principal component in the on pathway, and L2 is the principal component in the off pathway. So it's very likely that there's strong selective pressure for this coupling of these 2 postsynaptic elements abutting the same presynaptic element, although it's not known why. So we set out, for a variety of reasons that were not worth going into, to look to see whether DSCAM1 had a phenotype by Ian in a collaborative effort between my lab and the work of Sean Millard, a former postdoc in the lab, and Ian Montague, who now works in Canada and also at Genelia. And there were no phenotypes in DSCAM1 alone. There were virtually no phenotypes in DSCAM2 alone. And for also reasons that aren't worth going into, we looked at double mutants in mosaics. And Sean Millard and Ian and Juan Liu in Ian's lab carried out serial EM reconstructions of these mosaics. So invariant pairing is shown here. This is the photoreceptor terminal and the two dendrites, L1 and L2 in wild type. Invariant pairing, if you remove both DSCAM1 and DSCAM2, you lose that pairing, and you see many L1, L1, and L2, L2 pairs. So based on these observations and a lot of biochemistry and expression data using spliced traps, this is the model that we've come up with. Remember, these two processes of L1 and the two processes of L2 that I'm showing here are from the same cell. That's crucial. Each of these cells acquires a unique identity. For DSCAM1, it's through stochastic or probabilistic expression. Each gets a unique identity. For DSCAM2, it's for expression of two different isoforms in a deterministic fashion, where L1 always expresses the 2B isoform and L2 always the 2A isoforms. And that now makes them recognize self as self, and that's a repulsive interaction. And L1 and L2 repel one another, and this is permissive to pairing of L1 and L2, and obviously other types of cell recognition molecules must be involved in making this synapse. Now, DSCAM2 also functions in combination with DSCAM4 to regulate the patterning of yet a different dendrite, and I'll briefly show you that here. So here we're looking at the L4 axon here, and this is one of its dendrites, and it makes synaptic connections specifically with L2 but not L1. Problem we're very interested in, but we've made no progress in how that distinction is made. This shows the very simple dendritic pattern. This is the axon, and it has three dendrites. One that's very difficult to see here, that's the so-called home cartridge. This is the axon, and this little dot here is that dendrite. And then there's a dendrite to the cartridge that's just dorsal and just ventral to it, and you can see those as well. In a DSCAM2-4, so for DSCAM2 we found this by a candidate approach, and DSCAM4 came through a genetic screen unrelated to DSCAM2, so it was quite remarkable that both together, when you knock them out, give a much stronger phenotype. So DSCAM2 and DSCAM4 act together to regulate this dendritic patterning, a very precise defect. We've carried out a whole series of developmental experiments using a variety of different mutants, isoform-specific mutants, in various mosaics to look for autonomous and non-autonomous effects. And we've also followed this with live imaging. This is a summary of all of this in this model. So here, this is an L4 dendrite extending in. There would also be an L4 dendrite coming into this same group, and the target is an axon bundle at this stage. These are the medial side of the growth cones of photoreceptor neurons, and what we've sort of peeled away is a layer of glial cells that these growth cones are growing between. In wild type, they choose to target quite precisely then this bundle, and then they'll make synapses with L2 but not L1. And what we see in DSCAM2 for double mutant is that they no longer make contact with this initial bundle, rather extend out along the surface of these growth cones and extend to more distal columns. Now, based on these studies, we've argued that rather than a repulsive interaction, which is what we see with DSCAM2 and DSCAM1, that this is likely to be an adhesive interaction, where instead of being repelled from it, interactions are stabilized between this bundle and the growth cones of L4, and this then is playing quite a different role in regulating patterning of these dendrites. So in one case, in an L2 neuron, we know from genetic mosaic studies that L2 is required here in parallel to L1, and here it's acting, DSCAM2 is acting in an adhesive fashion, and yet, and what I described to you in the previous example is, and it's at a slightly later stage of development, DSCAM2 is acting in a repulsive way to mediate interactions between other processes within the same cell. So to summarize then, DSCAM1 regulates self-avoidance on its own. That's through a repulsive interaction. DSCAM2 regulates tiling. I didn't show you its role on its own, but it regulates tiling analogous to self-avoidance, again, through repulsive interactions. DSCAM1 and DSCAM2 regulate synaptic composition, and they do that through a repulsive mechanism, and DSCAM2 and DSCAM4 regulate dendritic targeting, and L4 targeting to different L2, L1 fascicles, and they do that through an adhesive interaction. Histological assays are really a drag. It's hard to convince people to do these histological assays. Phenotypes are generally fairly, in most cases, I showed you a good example where it was a very strong phenotype, but it's often quite a subtle phenotype. And you're quite limited in how many animals and lines you can screen. And so we were really very eager to develop behavioral screens. And we did a number of these screens in the early 2000s. And I'll show you just one, and it helped us identify an important component in regulating wiring, both within the lamina and the medulla. So this is a screen for R7 specificity mutants. R1 through 6 terminate here. They play a crucial role in motion detection, and R7 and R8 play a role in color vision, with R7 being the primary receptor for ultraviolet light. Tom Clandinin, in work I won't describe to you, who's continued this work in his own lab at Stanford, has done a beautiful work both through development as well as behavioral studies and circuit studies to understand motion detection itself. And he carried out screens for motion detection, and they were genetically mosaic screens similar to the one that I'll describe to you in a moment. Others in the laboratory, Herman and Ayosha Nernan, others set out to use a very simple color selectivity assay to identify wiring mutants. And in this case, they took advantage of an observation that was made many years ago, that if you take flies and you give them a choice between 350 nanometer light, so UV light, and 550 green light, they show this amazing preference or strong preference for ultraviolet light. And if you remove an R7 neuron, but leave the rest of the animal intact, they choose green light. So it's a powerful one. We reasoned that if you could just remove or disrupt the connections between R7 and their target, you would phenocopy this loss of the R7 neuron. And so we developed a simple way of generating, which was simple because others had generated the reagents that made it simple, to create mutant R7 photoreceptor neurons in an otherwise wild-type animal. It's really a great screen. It's the only screen that I've designed or was part of designing with postdocs that actually I think was a great screen. So the idea here was to make mutant photoreceptor. First, you make a fly in which none of the photoreceptor neurons are functional because they all express tetanus toxin in R7. So they're all wiped out. They behave as though they don't have R7 neurons. And if you then use mitotic recombination, you can now, in a small subset of those, at around 15%, you can generate an animal where 15% of the neurons have lost that tetanus toxin, and now that's strong enough to drive the ultraviolet response. So then we argued, great, if we could couple this with mutagenesis on that arm of the chromosome, we can now look for mutations that revert that, and they now no longer respond to ultraviolet light. And that was really a powerful screen, really was driven. It took a hell of a long time to do it, and Tori Herman really deserves the credit for generating that, and she's continued to follow up on many of these in her own lab at the University of Oregon. And it was a powerful screen, and you go chromosome by arm by chromosome arm. And I just want to show you one example of an important cell surface molecule she identified. And here, we just follow this up using genetic mosaics to look at the phenotypes using anatomical techniques. The green guys here are the control, and the green guys here are the experimental. And you can see that these are R7 neurons that typically terminate in this layer, have now terminated ectopically where R8 neurons would terminate. So this study, and this is an NCAD here mutant, and analysis of many other alleles that came out of that screen gave rise to this picture that R7 specificity relies principally upon NCAD here, and this is a very strong phenotype. 100% of the neurons mistarget to the R8 layer. And other cell surface proteins that are important in this process that came from the screen include LAR, two protein tyrosine phosphatases, LAR and P2P69D, as well as another cell surface protein that we know little about, a bunch of cytoplasmic proteins that are important, and nuclear proteins that play an important role in turning off the R8 targeting process in the R7 neuron. So NCAD here plays quite a specific role in R7 photoreceptor neurons, that's shown here. It plays no role that we can discern in R8 neurons. It plays no role in an L2 neuron, and then plays variable roles in other neurons. And the phenotypes that one sees are remarkably specific when you knock them out of individual cells. They have some commonalities, but sometimes these phenotypes can be quite different from one another. The screen I want to describe now is a molecular screen. And in part, it was motivated by this observation and fear that we were missing many genes because of redundancy. And so with the advent of RNA sequencing, we thought, great, we'll be able to look at all the genes that are expressed in these neurons, and we'll be able to get an idea of what we're up against. And so what I'm going to describe is an approach that we took to identifying specificity molecules in these 7 neurons, R7 and R8, and these 5 lamina interneurons. They all target the different layers in the medulla neuropil. They elaborate different morphologies, and they form different patterns of synaptic connections. Each of these is sort of smooshed together into a fascicle. It's shown here from the work of Genelia, from EM reconstructions, into a single column. And this column contains the processes of about 100 other neuronal cell types. So not 100 other neurons, but 100 other neuronal cell types. And that's repeated over and over and over again. Some of these neurons make connections with themselves, like L1 and L5, but to a large extent, they make connections with a bunch of other neurons. And these are the principal classes: MI neurons, DM neurons, and TM neurons. And remarkably, each of these then falls into on the order of 25 different classes, based on their morphologies and their patterns of synaptic connectivity. Now, I've attempted to summarize this here in the kind of slide no one should ever show, but I'm showing it for a reason, and that is, it is incredibly complex. If we look at the 7 neurons we're interested in, they're in green here and shown in green here in the schematic. Let's just take L2. So it is presynaptic to 8 different neurons, and that's what these are all designated here. And they are postsynaptic to 6 different neurons, and that's true for many of these other neurons as well. So an incredibly complex pattern of connectivity. And let me make one additional comment that specificity has nothing to do with the area of contact. That's an old idea in neurobiology, that the area of contact determines the number of synapses. There are plenty of neurons which have tremendous areas of direct contact from EM reconstruction, no synapses, and conversely, ones that have little contact, those contacts tend to be ones in which synaptic connectivity occurs. So the idea was to fact sort these neurons, do RNA sequencing, identify the small number of cell surface molecules that would be expressed in them and play an important role, and solve the problem. So the first problem became clear when we looked at the degree and number of cell surface proteins expressed in these neurons. And this is just a quick summary of that. The 7 neurons shown in each column, each row represents a different cell surface protein, and each of these neurons expresses a large number of different cell surface proteins, between 250 and 325, about 976, so a quarter to a third of the cell surface proteins with relatively high RPKM values. Lots of differences between any pairwise combination, and few differences that are highly specific, meaning on in one but not the other 6. So each neuron has a unique set of cell surface proteins, and far more than we were hoping would be the case. We became particularly interested in one family, and they're shown here. This is DPR proteins. They are 2Ig domain-containing proteins, and their expression pattern in these 7 neurons is shown. And many of these have been verified using a mimic technology, which is simply a protein traps. And the vast majority of what we see from RNA sequencing holds up with these traps, which is very encouraging. So what are DPRs? The first DPR was identified by Craig Montel in a behavioral screen some years ago. And the remaining 20 paralogs were identified by homology. In a beautiful biochemical screen, Engin Özkan in Chris Garcia's lab identified a group of interacting proteins. The DIPs have 2 Ig domains. The DPRs have 2 Ig domains, and the DIPs have 3 Ig domains. In the original publication, there were 11. That came back down to 9. And through collaborative work that we carried out with Larry Shapiro and his colleagues at Columbia, we're back up to 11. So this is a little outdated. We only have the 9 here, but it makes the point. It's complicated. DPRs interact heterophilically with DIPs. It's not a one-to-one correspondence for most of them. They can interact with multiple ones, so multiple DPRs for multiple DIPs and vice versa. DPRs can interact with themselves. They interact with other DPRs, and DIPs can act homophilically. I don't know of any examples where they can also act to promote interaction between different DIPs. So a very complex network of interacting proteins. So I told you that the DPRs are expressed in this cell type specific fashion. It's also quite dynamic within the L1 through L5 and R7 and R8 neurons, but we found no DIPs that were expressed in this way. They're expressed in the targets. There are, as I said, 11 DIPs. We have reagents for studying 9 of them. We'll get the other 2. And we've done the analysis of 8 of them, and I've shown you the expression of 6 of them here. And what was really striking to us is that they're expressed in a layer-specific way. This is using protein traps. We've been looking for years for genes that would be expressed in a layer-specific way. So this was quite surprising to find this and gratifying. We unfortunately can't, so let me just simply say, they come on in layers. They largely stay on in the same layers throughout development. The DPRs, by contrast, are quite dynamic in their expression. Some stay on in a cell, others vary, go on and off during development. You can't determine, because of the density of different neuronal cell types, you can't tell which neurons are expressing a given DIP by this kind of analysis. And to do this, we turned to a method called the MCFO method that was designed by Alyosha Nern at Genelia, a former postdoc in my laboratory. And this shows its power. It's sort of a rainbow, it's a derivative of rainbow, if you will. So this is one DIP that GAL4 has been inserted to, and it drives UAS-GFP. You can see it's quite broad. There is a layer of specificity in this expression. You certainly can't tell what cell is expressing it. The way to do this is to now make a line that has 3 UAS constructs. Each of them has an interruption cassette, and they're off. So in the absence of flip recombinase, there's no expression. And then with limiting levels of flip, you can get the occasional one to flip. Each one is GFP, which has multiple copies of an epitope, so many copies of the epitope that you wipe out the endogenous fluorescence, and you just use immunodetection to determine the pattern of expression. And you get this lovely kind of rainbow-like brain, and you can begin to sort out who is expressing which DIP based on its morphology. So this is a neuron that plays a role in the off pathway of motion detection, called T4. Another one called the DM18 neuron, which has this beautiful broad arborization through about half, very large extent of this layer in pink. So by comparing this expression to the biochemistry and to the connectome, we found that many of these DIP-DPRs are expressed in synaptic partners. So I want to summarize that here for one neuron that I'll describe some functional studies for in a moment. So L3 makes over 10, has synaptic connections with over 10 partners. And that's shown as these arrows, with the arrow indicating the direction of synaptic transmission. Red indicates the DPR or DPRs rather, that are expressed in L3. And there are multiple DPRs expressed in L3 in a dynamic way. By contrast, the neurons in the medulla, and virtually all neurons in the medulla, express a DIP. And typically it's a single DIP. The exceptions turn out to be paralogs that are recent duplications that have largely the same binding specificities. So here, for instance, DIP alpha and DPR610 interact with one another. They're expressed in synaptic partners. And what I'm going to describe are some genetic studies that we've undertaken to get at the function of DIP alpha and its cognate high affinity interacting proteins, DPR6 and 10. So just a little bit of information on DM4 and DM12. These are amacrine-like cells. They extend into the medulla and they extend out within a layer. They both extend within the M3 layer, and they have largely overlapping synaptic connections, with most of their synaptic connections being to L3. DM4 neurons tile. As a consequence, an individual L3 neuron makes synapses with only a single DM4 neuron. By contrast, DM12 neurons overlap extensively. There's a threefold overlap. So a given L3 terminal will make synaptic connections to typically to 3 different DM12 neurons. And in the discussion, we can come back to why that may be significant. The right-hand panel shows the relationship as determined by Alyosha Nern and his colleagues between these terminals within the column, where you have, if we're looking down on it, the lowest layer is within the layer. The lowest layer of the processes are, you have this horseshoe, if you will, of DM4 terminals, and then lying on top of that you have L3, and then DM12 is on the sides. Okay, so let's look at what this interaction does in these neurons. So first, proteins generate, antibodies generate to DPR10 and DIP alpha, as well as DPR6, which I'm not showing you here, are expressed in the incipient M3 layer. That means an early stage of M3 layer development when these processes first interact with one another. And you can see that this is when DM4 is extending its process within this layer. There's DM4 process here as well as here, and DM12 is quite similar and grows in about the same time. So they're expressed at the right time to be doing something interesting. This is long before synapse formation, just as these processes grow into the neuropil. This is another example for students of the kind of slide you should never show. Okay, and I'm not going to drag you through this, but just to remind me that we've used two different genetic techniques that allow us to identify individual cells by their morphology, and in those cells and only in those cells, count the presynaptic release sites. Okay, and this comes from the work of a variety of different labs, and we sort of put it together. Okay, so I won't take you through this other than to say that we can generate mutant cells or control cells that turn red, and the green dots are their presynaptic release sites. And we can do this on the postsynaptic side as well. So let me show you this for DM4 and DM12 here. The left side shows two panels, a wild type, DM4, and mutant, and you can see that there's a reduction in the size of the extent of process outgrowth within the layer. So instead of covering about 16 columns, it covers 11 columns on average. DM12 is different. So DM12 covers about the same number of columns as DM4. By contrast, in these single mutants, about 60% of them, I should say all of them, have about the same coverage within the M3 layer, but about 60% of them send a process to a deeper layer where they then arborize, and that's a defined layer. It's always the M8 layer where they arborize. And also, you can see these little puncta indicating that they do elaborate presynaptic sites. We don't know what they synapse on, however, in that layer. So we set out to ask whether this requires heterophilic interactions with DPR6 and 10, or the Shapiro lab had discovered that, regrettably, DIP alpha not only binds heterophilically, it also binds homophilically. It binds to itself with a slightly different KD, but not so slightly different. It's about 10-fold difference, but it's certainly significant. And so we made two types of mutations based on the crystal structure from Eng and Ozgen for the DPR6 DIP alpha, which is really the same as DPR10 DIP alpha, and we used the structure that Philip Kosminescu had determined for the DIP alpha and homophilic interaction. So we knocked these mutations into the germline thanks to the enormous power of CRISPR, and we made the heterophilic mutation, and I won't be talking about this heterophilic mutation in DPR10, just the DIP alpha heterophilic mutation is I83D, and we made this, we did the mosaic analysis the same way I described before, and its phenotype is identical to the null, so heterophilic interactions are required. By contrast, if we make the mutation that knocks out homophilic binding, and I should say the heterophilic and homophilic binding deficient alleles, the proteins encoded by them are expressed at wild type levels, we see no phenotype. So clearly heterophilic interactions are required. So we then asked, is interaction between DPR10 and DIP alpha sufficient to promote targeting to a different layer? So if we expressed DPR10, for instance, in a different layer, could we get DM4 or DM12 to go to that layer? And this is the way we carried out the experiment. We being Shishao, Liming Tan, and Shua Zhu. So here, this band represents the incipient M3 layer. So we began by making a double mutant that removed both DPR6 and DPR10 from that layer. And then in that genetic background, we targeted expression to a layer that's just below it early in development when these layers are really close to each other. And then during subsequent development, the good news is if you're trying to spur phenotypes is they separate out because there's intercalative growth of other fibers and it pushes them apart. And you can see that there's really quite striking effect on DM12. If we now misexpress DPR10 in this layer, we can see now that these fibers project to the deeper layer. That's also true for DM4. And this is dependent upon an interaction with DIP alpha, because if we do this misexpression in a triple mutant removing DPR6, DPR10, and DIP alpha, or the DIP alpha heterophilic binding allele, we abrogate that mistargeting. So from loss of function and both gain of function, it's clear that DPR10 DIP alpha interactions are important in some way for regulating the interactions between these processes within the developing M3 layer. So let me just briefly make a comment about synapses. And obviously this needs to be verified and analyzed in detail at the EM level as well, but we have not yet done that. This shows that for DM12, regardless of whether they target only to the M3 layer or whether they go to M3 and M8, they show this reduction in M3 of about 30 to 40% reduction in the number of presynaptic sites. Of course, there is no, the DM12 neurons don't project to M8 and wild type. There are no synapses there. They do, however, synapse on something. We're not sure what they're synapsing with in the M8 layer, but they do form presynaptic release sites at that layer. We also see that there's probably a slight difference. It's not clear on the lateral extent for DM12, but nonetheless, the density of synapses for a column is reduced by about 30% versus wild type. By contrast, DM4, remember, has this 30% reduction or so in the extent by which they extend within the layer, and we see a commensurate reduction in the number of synapses. But the density of synapses per column is the same. So to summarize then, DIP alpha and DPR610 interactions required for target layer interactions in M3. DIP alpha DPR610 is required for synapse number in DM12. We don't know how that works developmentally, but that is quite a clear result. DIP alpha and DPR610 also regulates cell number. There's a modest reduction in DM1 and DM, which I haven't talked about, which it plays a role in M1, where DIP alpha and DPR610 are also expressed, as well as for DM4 and DM12, and that by antagonizing apoptosis upstream of the HID protein, and in work that I don't have time to talk about today. DIP gamma and DPR11 regulates interactions in M6. That was first shown by Chris Garcia and Kaizin and his colleagues, and it's a bit controversial. I'll be happy to talk about what those effects are, but they clearly do interact in the M6 layer. We've also explored that in some detail as well. So it seems as though different DIPs and DPRs are playing roles. It's been complicated to get at those roles genetically, in part because of redundancy, but clear examples for DM4 and DM12, and also for DM8 neurons, which express DIP gamma. And we continue to analyze the other DIPs and DPRs in various mutant combinations to gain more insight into what they're doing in establishing these patterns. It's always disconcerting when you realize that you've gone longer than you should. It's also disconcerting on this panel that you also get the time of day in LA, and it's 2:54 a.m. So I'm a little slower than I normally would be, but I'm not going to drag you through this next slide, which is a summary slide of what we know about other neurons that target to the M3. Simply to say that other mechanisms are necessary for targeting of other neurons to the M3 layer. So they don't all use DPR6 and DPR10 interactions. They use other mechanisms. They use netrin, they use, depends on the neuron, they use semaphorins and then cadherin, and that's only looking at 4 different classes of neurons. And my guess is that the many other classes use some of those and other mechanisms to get to the M3 layer. In the last little bit, I want to turn to what was really a surprise for us, and that is that we've uncovered really robust spontaneous activity in the developing medulla and lamina. In fact, an entire fly brain that occurs with the onset of synapse formation. And this came about from really a wonderful work carried out in my lab by Ilkin Akin, who developed a very powerful way of doing live imaging to study axon guidance. And he came into the lab to study a specific aspect of axon guidance. An MD PhD student came into the lab and said, hey, this is really cool. I'd worked with GCAM6S as an undergraduate. Maybe we should just put that into these neurons and see what happens during axon guidance. So they did this. They did it for the R8 neuron. They watched what happened during axon guidance, and of course there was lots of stuff going on, but nothing particularly interesting. That is, nothing that happened at a particular time in the trajectory of the neuron. But they just left it on the microscope. These are fully intact flies. They actually close and walk away during the imaging when they're done. So that's when the experiment ends. And What they did note is there was this very robust spontaneous activity. Now, spontaneous activity in the developing nervous system is part of the canon of vertebrate neuroscience. And flying neuroscientists have largely said, it can't be that important because we don't see spontaneous activity, largely don't see spontaneous activity in the developing central nervous system. Of course, part of the reason we didn't see it is nobody looked, okay? Because it wasn't an easy way to do that. And so this wasn't why we set out to do the experiment, but as fate has it, it introduced a whole new way of beginning to think about this problem. And I'll take you through this quickly because we don't actually know what it does, okay? So the bottom line is it's everywhere. Different neurons have spontaneous activity. Each neuron has a particular pattern of spontaneous activity, and yet it's synchronized and it's coordinated in some way that we're trying to understand. So here is a sort of a broad example. In fact, you can put this under a dissecting microscope, and you can see these patterns of expression, these patterns of activity if you have enough, you've doped it up with enough of a transgene of GCAM. So here we have on the left just GCAM expression in a static image. And this just shows GCAM is expressed in all neurons here. This is the medulla, and these are the two deeper neuropils, the lobe and lobular plate. And this is about 6 minutes worth, sped up 25 times. Okay, so lots of activity. Some look like waves, some look like they might be spontaneous. Remember, it's a mixture of a ton of different neurons here. Okay, so let's look at this a little more carefully. This puts the activity in the context of sort of a broad description of development of neurons in the visual system, early differentiation, axon guidance, beginning in late third instar development and proceeding into pupil development. And then this activity begins with the onset of synaptogenesis, and it's long before vision. And we know from mutants it's completely independent of any sensory input. The activity begins a little earlier than I'm showing here. It begins about 50 hours, and you see little traces as though something's warming up to happen, and then it sort of just takes off, and then you can see it sort of planes out. It begins to decrease and is gone by closure. And then, of course, the nervous system is responsive to visual input and all sorts of other kinds of inputs as well. So we'll delve a little deeper into this. This shows what these periods look like. We divide what we call a period into two parts, what we call an active time and a dark time. So here's an active time. Here, followed by dark, and so on. And you can see that there are multiple peaks within any one of these active times. This is for the whole brain. Okay, this isn't for individual neurons. So let's take a look at individual neurons. We've done 15 individual neurons, and I'll begin with 2 of them. I won't drag you through each of the other ones. I'll summarize them at the end. So these are 2 neurons, TM3 and L3 neurons, and they have different patterns. Now, I want to emphasize that these patterns are coordinated. When neurons are active, they're active at the same time. And when they're quiet, they're all quiet at the same time. And I'll add that during that quiet period, that's when the glia are active. So there's something probably quite interesting about the relationship between glia and neurons in this system. So let's take a look at what TM3 looks like. And I'll turn off L3. So this is very wave-like. So these are firing away, and it's silent. And then it begins firing away again, and then silent. And we'll look at one more round, and then we'll turn it off. And it just keeps going. This is L3, which is different. It's highly synchronous. So same periodicity. So it's active, and then it's quiet, and then most of them seem to be firing at the same time. We'll look at one more round. So these are 2 quite different behaviors. And so we sought to quantify this in part. So one of these looked wave-like to us, and the other one looked like it was synchronous. And we realized as we began to look at more of these that we needed some way of quantifying this. We need some metrics by which we could compare activity. And Orkun came up with this very nice way of doing that. And that was first just to take the average intensity across the field, and then to look at that at each time point. We end up getting these kind of traces, and of course this TM3 TM3 and L3 trace look awfully similar to one another, so we can look a little bit more closely at them. Let's expand just one of these active regions, and now you can see each of them has a substructure. And we can analyze these substructures by just generating chimerographs for them. So here we have time. Remember, this one active unit is about 6 minutes or so. And we have time then along the horizontal, and we have the position of the columns along the vertical. And we're going to define two metrics here, or Kuhn defined two metrics here, one called coordination and one called coherence. So if we look at each one of these peaks, which Kuhn has dubbed sweeps, so that's shown by these blue lines, that each one of these is a sweep. It's made up of individual firing patterns as well, but each of these peaks is considered a sweep. And these sweeps have different structures. You can see that here for both TM3 and for L3. So let's just take a look at this guy here on the far left. You can see that most of the columns are firing within this sweep. By contrast, this one from the second from the right, just a few of them are firing. By contrast, most of the ones for L3 look similar, and that we probably would have come to from qualitatively from looking at the slide that I showed you earlier, their activity. They all, or the vast majority of them, are firing at the same time within a given sweep. And we call this behavior coordination. It's a measure of column participation per sweep, the fraction of sweeps that have greater than 90% participation. Kuhn then defined a second metric, and that is the position of the peak, okay, for each of the columns within each individual sweep. And he called this coherence. And you can see differences in the coherence of TM3 and L3. You can see that the peak for each column spans a broader time frame than what we see for the peaks in L3. And he called this activity coherence. Okay, so it's a measure of the peak time spread. It's a fraction of columns with peak at the same time. And then we can plot each of these separately. Each circle represents the average value of coordination or coherence between 55 and 65 hours from a single time series, and the bar represents the average. And here we've arranged them. This was L3, TM3 and L3, and then the remaining 13 are shown here, and they've been ordered by coordination. You can see that there's no clear relationship for each neuron, or a consistent relationship between coordination and coherence. They're different types of metrics. We can plot these against each other, and we end up with this plot. And the purpose of this plot is really twofold. One is it provides us a way of comparing manipulations and the effect on the activity of different neurons. And they also allow us to compare this activity to the adult connectome. So for instance, this group is really interesting because L3 is presynaptic TM9 and DM4, suggesting at this early time that there's an early version of the mature connectome. And we actually know from some optogenetic studies that we've just begun that indeed some of these synapses are forming at a relatively early stage during this period of activity. And I'll be happy to describe how we do those experiments in the discussion. These two are also synaptic partners, L5 and MY1. Others, which are not synaptic partners, are actually quite far away in these maps. So suggesting that at this early time, if they do have synaptic activity, this is due to synaptic activity, it perhaps is with other synaptic partners. And it's well known, or there are other examples, one from C. elegans that I'm aware of, and a number in vertebrates where neurons have temporary targets with which they make synapses. That's not the mature synaptic specificity. They make immature synapses, and then they change their synaptic specificity with maturation. So we're in the process, obviously, of trying to figure out what these do, but it raises a really interesting possibility that layered on top of what I've described in terms of molecular interactions that are important in wiring, that there's this other time scale of interactions occurring at the level. We have some data that this is due to synaptic transmission. May also be playing an important role in sculpting these activities. finally, let me just summarize. There are multiple processes regulating specificity. It's an incredibly complex problem. We were dismayed to find that there were hundreds of cell surface proteins that are expressed in neurons. And I described one time point, but I should say we've done time courses for a variety of different classes throughout the process of synapse formation of any specific neuron. And we see massive changes in the dynamics. And these are large changes, not only in number, but in magnitude of expression. Whether that's important, still remains to be determined. There's a core set of cell surface molecules that are important in regulating interactions between neurons. Cadherins, netrins, semaphorins, they all play a role in wiring the five visual system. And I give you a few examples of that, but there are many more. In addition, there's duplication and divergence of families. Of course, the DSCAM family is quite large, plays a role in self-avoidance and tiling, and undoubtedly in other processes we've yet to uncover to regulate patterning within this complex neuropil. And we've recently uncovered this spontaneous neural activity, which suggests that there's another type of cellular communication, which is also important in this process of getting these synapses correct. And I want to just leave you with some feeling for this, the complexity of the problem. And to me, this was really driven home by the work that's been done on the connectome. 100 plus neurons in each one of these columns. Each one makes synapses with probably on average anywhere near 5 to 15 different targets, pre and post-synaptic. And the specificity is extraordinary within a given column. It occurs over a long period of time. I think one might expect that this kind of complexity is necessary to discriminate between all the possibilities that a neuron has for making synaptic connections. This level of complexity is very similar to the vertebrate retina. My guess is it's similar from recent sequencing studies, probably not so different than the cortex, which has about 100 different cell types in the visual cortex. So I think there's lots to do, and I think we also need new ways of thinking about the problem and thinking about this complexity. Clearly trying to understand what the genetic programs are, how they're coordinated, and what are the principles underlying these programs. And I think the tools are marvelous for getting at those sorts of problems. So finally, let me just thank a really talented group of people who've done the work that I described today. And of course, much of this work relied on people that I have not acknowledged here who had worked earlier. What I showed you with DSCAM1, DSCAM2, double mutants was work by Sean Millard when he was in my lab, and he's continued to do this work in Australia in his own group, with Ian Myatagan and Joa Liu, both of who continue to work on this, both in Canada and also at Genelia. Will Tadros carried out the work on DSCAM2 and DSCAM4. The DPR and DIP work was carried out by Li Ming Tan, Qi Xiao, Shua Zhu, and I should say Li Ming has gone on in the lab to venture into visual cortex to see whether we can deal with something even more complicated. And in the early work, Zhu Yanyu and Ying Lin carried out some of this work, sort of cut their teeth on this project, and then went on to consider other projects, work on other projects involving wiring specificity. We've done a lot of transcriptomics, and I've talked a little bit about the work of Kelvin Zhang, Matt Peiko, Will Tadros, and Li Ming was a real driver of that as well. And that work was started by Marta Mori when she began in my lab and worked on R7 recognition specificity. We've had a really fun time working in collaboration with Larry Shabiri and Barry Honig's laboratory, in particular Philip Kosminescu and Alina Sergeyeva, in looking at the DPR and DIP family proteins through crystallography, as well as biochemical and biophysical studies. This work involved a tremendous number of different reagents, and we are really tremendously indebted to Hugo Bellin and Sonal and Painting from his laboratory, and a lot of great collaboration and communication, I should say, with Kaizen and Caltech. And we've been beneficiaries of the EM and markers and reagents that have been generated in Genelia over the past decade. So thank you for your attention. I'd be happy to take questions.